Hello, my name is Bridie Alexander-Lawrie. This is my research, recombination and inheritance in mitochondrial genomes of a freshwater gastropod. See down here. So, what are mitochondria? You may have heard of them. They're throughout your entire body. They look like that guy over there. So, they're an organelle in all your cells. They are the main energy producers. They originated from an endosymbiosis of a alpha proteobacterial cell into the ancestor of eukaryotic cells. They currently are an organelle. Their impact on the cell, their core to energy production, as I said, dysfunction in them naturally progresses into disease. And if you look down here, these are some of the enzymes that are in the mitochondria. The mitochondria is basically all cell wall, right? Membrane. And so it has these all across it. That's how it makes energy. There are huge ties with the nuclear DNA and the mitochondrial DNA. So it was the alpha proteobacterial cell was endosymbiosed. It has its own circular genome. So you have mitochondrial DNA, which is mtDNA, and you have nuclear DNA. If you look at this enzyme here, the purple here is what's coded by the mitochondrial DNA, while this white is encoded by the nuclear DNA. And you can see, pretty intricate. It's all got to connect together. So it's got these huge ties to nuclear DNA. So if you had a huge sequence divergence in your mitochondrial DNA, you really wouldn't have these functioning enzyme complexes, right? They'd be unable to work. So it's really important that you have low sequence divergence of mitochondrial DNA. So it matters where you're getting your mitochondria from. Who are you inheriting it from? So I was recently a writer on a paper that's currently under review called Inheritance Through the Cytoplasm. And over here we've got this lovely graphic showing mitochondria and chloroplasts. Both of these are plasmids that are inherited through the cytoplasm. And this image here shows you how cytoplasmically inherited elements can produce variation at all sorts of levels of organization, right? It's not just individual little organelles that change, it can change the whole plane's function. Like I said, this is how this function can lead to disease. So there are two main categories of inheritance: uniparental and biparental. So this is uniparental. This in particular is maternal inheritance. As you can see, all of the offspring are having the exact same mitochondria as the mother. There's no combination here, just passing down linearly from mom to all offspring. And over on the right of the mitochondria, the two lines, that's the nuclear genome. So it's combining following Mendelian inheritance, whereas the mitochondria is being inherited non-Mendelian. However, this is biology. No system is perfect. So even in this maternal uniparental inheritance, you sometimes have some not perfect inheritance. And so you have what's called heteroplasmy. So hetero means different, plasmy means plasmid. And heteroplasmy is when cells, or even an individual mitochondrion, contains more than one type of mitochondrial DNA. So I've got two options here, right? This is a cell having multiple mitochondria, they're from different genetics, or one mitochondria having different genetics within itself. And the definition heteroplasmy kind of implies that it's not balanced. So this is often called leakage. It's accidental. It's if it's maternal inheritance, you have paternal leakage. And then that brings me to biparental inheritance. What if it's balanced? That's what biparental inheritance is. So as you can see, kind of pass down how you would think it maybe you would be. So you have the maternal and the paternal. They're either together, like we saw in the other cell, or they're merged. So you have one mitochondria that has multiple genes. The token species for this is yeast. Yeast actually has a big matrix, which you can see over there, where when it's first a zygote, it has the paternal and the maternal on different ends of the matrix, and as it grows, they start to merge, recombine, and kind of conglomerates into one. And then, of course, yeast is isogamous, so you don't have female male, you have A and alpha. So there's a specific version of this called doubly uniparental inheritance. It's kind of a subcategory of biparental. And it's really interesting because it's pretty stable. So what we have here is the maternal, their maternal genome, and the paternal is carrying a paternal genome and the maternal. So any males are carrying 2 mitochondrial genomes. All the females are inheriting uniparental. So the daughter of your mother is going to have her gene. However, the males have this paternal genome that is passed down through the gonads. Forgive me for not including that in my diagram. And that's one way that it keeps it pretty separated, right? Because if you have all of your mitochondria located only in the gonads, you can they can hunker down there until reproduction happens. So far, this has only been seen in mollusks. This is a table from my paper that's under review, just showing a few different species that have some alternative inheritance mechanisms. That means they're non-uniparental. Uniparental is seen as the norm. But anywhere that you look for it, you kind of find this divergence. So here we have like the mouse, paternal linkage, it's a small percentage paternal inheritance, opposite of maternal inheritance, right? And then biparental inheritance. So, why Potamopyrgus antipodarum? This is my snail species. It's a gastropod that's native to New Zealand. It's hugely invasive everywhere else across the world. And so, like I said, mollusks often take part in DUI. This is often because of the stability, right? If you're locating them in one place, they don't have to worry about recombining and making that high divergence. So far we've only seen it in bivalves, but the patterns that we've seen of this DUI inheritance are likely ancestral. The evidence that we've seen suggests that it probably occurred before the split between gastropods and bivalves. So, hence, I'm looking at gastropods. P. antipodarum is an upcoming model organism, and that's often due to its variation in sexuality. So you'll have sexual organisms and sexual lineages, and you'll have asexual organisms and lineages. They're not hermaphroditic, right? Meaning they have one set of genitals or they're asexual. And this asexuality allows us to have more like pure lineages and more separation between the lineages. So over here, I have a map of the sexual and the asexual variants, and you can see how they vary from in different parts of New Zealand. So another reason that this is my species of study is that we've seen some really odd patterns of SNPs in pooled reeds. So this chart here is the genome. Mitochondrial genomes are typically around 17,000 base pairs. So these are all the different positions on the mitochondrial genome. So it's not unusual to have a couple different versions of SNPs, right? We have GA and AC, or GA and TC in this case. And the weird thing that we've been seeing is that while we have like it's reasonable for us to have these two different SNPs. What's unreasonable is for them to be recombined. The recombinant mitochondrial DNA suggests that there's heteroplasmy, right? There's somewhere where these mitochondrial genomes are combined and exchanging information, which they shouldn't be able to do in maternal or uniparental inheritance, right? Because you only have one. So this implies heteroplasmy or biparental inheritance in this species. So that's my big research question. Does P. antipodarum exhibit biparental mitochondrial inheritance and or mitochondrial recombination? So here's my design. I'm testing for heteroplasmy, right? In order to see how this is happening. How is this recombination? So I'm starting with a breeding design, followed by husbandry and sexing, DNA extraction and mitochondrial enrichment, a pooling strategy and sequencing, and then ending out with extracting leads, mapping, and analysis. So step A, breeding design. We had some snails crossed at the University of Iowa, and these are different, over there on the map, you can see the different lakes. These are different species from those different lakes. And they're different, they're mitochondrial genomes we've looked at. We know that they're different enough that with these crosses, we should be able to see heteroplasmy. However, between Alexandrina and Selfie, they're a little too close, so we're not crossing those two. And we can still get good information from these two crosses. We are also selfing our individual, I mean, this is selfing, but we're selfing each of these lakes within their own populations, and that's going to be our control. So, step E is the husbandry and the sexing. They're kept at 16 degrees Celsius in carbon filtered water. That's what they like. Over here, I just have a diagram of what we're exactly looking at. They're long snails. Comparatively, they're very small. But we fed them spirulina in the same carbon filtered water. But this is a diagram of the sexing down here. So we painted them on this underside of their shell while we were sexing them. And this is just a diagram of how we were sexing them. So they're not hermaphroditic. One of them has a penis, the other one does not. It has a rooting sac on its neck. And over there is an actual picture of the snail flipping over. You motivate them to flip over and do sex them. Next, we did the DNA extraction and the mitochondrial enrichment. So we got our snails, we cracked them out of their shells. We put them into chaos and EME to lyse the cells. We then waited 2 to 10 days to make sure all the cells are lysed. We added RNAs so that we don't have to worry about RNA data. Then we ran them through a phenylchloroform DNA extraction. And that gets rid of anything except for the DNA. The ethanol precipitation separates it even further, purifies it. Then we did a double restriction enzyme digest. So that's on a low melt gel that we're running it. And basically these two enzymes that we're using, they're taking, because we still have all the DNA together, right? We have the nuclear DNA and the circular mitochondrial DNA. So our two restriction enzymes are snipping the mitochondrial circle into a line and getting all the nuclear DNA and just cutting it up into all these little tiny pieces. So when we run it on the gel, we end up with these two clear lines. So up here is our mitochondria, right, at 17,000 base pairs. And then down below, we get this big old smear of nuclear DNA that's all being cut to different lengths. And that's what one of the restriction enzymes does. The other one actually just makes the separation even further by cutting the nuclear DNA. So then I cut out these top bands with a razor blade, put them through a gel exclusion column, and then we have our mitochondrial DNA. It's a mitochondrially enriched DNA, right? It's not going to be pure. Here I have it as a circle, it's a line, right? Just for clarity because it's a plasmid, right? So then we had to do our pooling strategy and actually run our sequencer, right? So this is just the results we kind of got. I have DNA extractions from 100 snails. Currently, the females are around 20 to 50 nanograms per microliter, and the males range from 8 to over 100. So we're expecting, as I do more of the females, like quantification, that we should have between 10 and over 100 nanograms per microliter for all of our different snails, 100 snails. So with my pooling strategy, I'm pooling by parents and cross number, right? Because we did each cross a number of times. So this is my notation strategy for that. Basically, we're starting with the maternal link, then the paternal link, and then the cross letter. And it's, cross A, cross B, cross C. So over here, I have AMC. That's Alexandrina, is the maternal link, Maurika, the paternal link, and it's cross C. And so this is how we pool them. We pool them by the cross and who their mother is. And these are the individuals that I got for each of these male crosses, which is one that we've seen. So, sequencing. So this is a very easy drawing of what's going on with the barcoded adaptive ligation. We put barcodes on each of our individuals in our pools, or each of our individuals in the pools, so that we can identify them later. So it's not as easy as just putting an adapter on. Realistically, you have to fix the DNA. Then we put an A on either end that allows us to adapt on the barcodes, and then we ligate on the adapters. And the adapters are recognized by the nanopore so that it can read it. It can grab it and read it. This is what it looks like. When I keep saying nanopore, it's Oxford nanopore, which is next generation sequencing. It is a living pore on a chip, a computer chip, and the DNA flows through, and as it goes through the chip, it releases a electric discharge, which allows us to tell which nucleotide we're looking at. Which is a really cool system because you're literally just reading it like a strip of DNA. You don't have to cut it up. So we can do this long read sequencing really easily. And then the analysis and the export. This is what it looks like. It's a bunch of twinkling lights because some of them are working at one time, some of them are working another. So it's very cool. Then we have to extract our reads, map them, and analyze what we're looking at, right? This all looks very physical. It's all happening inside of the computer, our coding. But basically, we'll have our so we read through, we have our F and our P generation DNA reads, right? Mom and kid. And then we're comparing them to the mitochondrial genome that we have, the reference mitochondrial genome. And that way we can separate any nuclear reads that manage to get through our enrichment. So we already sent our female reads over to NCGR, where they were assembled by Evan Lavelle into our reference mother mitochondrial genomes for every mother. And that was funded by InBred. Then, so we have our mother reference, and we're going to compare all of our male F1 to that reference. And what we're looking for is we're looking for SNPs, or single nucleotide polymorphisms, to see if there's any divergence. If there's divergence, it's implying that heteroplasmy. And if we see that heteroplasmy in the crosses versus the versus our control, we can kind of tell where that heteroplasmy is. So we've already sequenced the males. These are our sequencing results. We have a ton of breeds here, some more than others, but we have plenty to deduce whether there's heteroplasmy, why there's heteroplasmy. Because this coverage is crazy, because we had, on the low end, like 0.7 times, but on the high end, we had hundreds of covered breeds. So that's hundreds of breeds of the mitochondria. So, in summary and future directions, our snails were bred in the University of Iowa by Maurine Neiman and all of her undergrads. Our DNA has been extracted for all of the snails. Mitochondrial enrichment has been done for the males, but I need to do it for the females. Sequencing has been done for the males. Extracting the reads, mapping, and analysis. It's in progress on RMAC Summit, which is a supercomputer. And all my scripts have been written. I need to figure out, there's kind of a lower yield with the first bead cleanup if I go back to this. So over there, with that first step, in between each step, I need to put the DNA into beads and do bead cleanup to get rid of any proteins. And in the first one of those, I'm getting slower amounts, but ideally we can get higher yield. But the reads that we're getting, we're still getting a lot of coverage. So we'll be able to see that here. So I'd just like to say thank you. I have my funding up here on the left, New Mexico Tech, NSF, New Mexico Inbred, Cytonuclear Lab, of course, Joel, my advisor, Aaron, doing all of the background things that are so necessary. Cameron, Maya, Emily, and Link, also helping with the snails, along with the rest of the lab. All 100 plus snails that have died for this cause. The inheritance in the cytoplasm crew with Laurentia Camus and Greg Hurst over across the pond, writing that very long paper that is under review. The Neiman lab, of course, for collecting all of the snails and sending them all over, doing all the process for us. Evan Lavelle with NCTR, mapping the mothers and the mother mitochondrial genome. The superb snail photography done by Bart Zielstra and general support. Yeah, is that right? And then general support, my partner, Karen Gantz. My partner, Sylvia, Karen Gantz, for just supervising the general support. And then my roommates, Adrian. Any questions?
Joey has a question online.
Okay. Yeah, go right ahead. I hope I can hear you.
Hi, can you hear me?
Yes, yes, great.
Great, nice presentation, Bridie. I have two questions. I guess I can ask one of them. One is more like maybe theoretical, I don't really know, but you mentioned DUI, double parental inheritance, and I was just wondering, it's only seen in these mollusks, I guess, and is there an evolutionary advantage to that? I don't know, I'm just curious for your thoughts.
Yeah, that's a great question. So as far as only having seen, I can go back to we were talking about that. As far as only being seen in bivalves, that's the only place we've looked for it. So it could easily be in other species, but we haven't really, done the searching for it. As far as evolutionary advantage, uniparental inheritance can get really problematic because if it's working perfectly, you've got a perfect system. It's kind of the thing with a lot of inbreeding where it doesn't go very well. And already, like the mitochondria has shucked, I think like 90% of its genome over to the nuclear genome. So, it's a very small amount of DNA. It's 17 base pairs or 17,000 base pairs. And so the less, like a small amount of recombination is necessary, right? So if you're not getting any of that, you have kind of inbreeding like problems. If that answers your question. So like DUI would have the advantage of, there's this interface between the paternal and the maternal where you can have some genetic recombination, and that can be beneficial for the mitochondria.
Great, thanks. And if no one else has any other questions, I can always ask one, but thank you.
Yeah, totally.
All right. No one else looks like they're asking anything. So, sorry if I cut someone off. But the second question is more related to methods. You mentioned your long read assembly was done by Evan. Nice guy. And I was wondering if you know what assembler that he used to do that, like, I guess,
I don't actually.
long read mapping.
Is the Canu assembler, which is the Solera assembler for long reads.
Okay. It's a, yeah, he did do
Canu,
that.
C-A-N-U, Canu.
Yeah, the Canu assembler. It's
Okay.
like, yeah.
It's the, so Solera is Craig Venter's group. Craig Venter is the guy who tried to privatize the human genome.
Oh, great. Yeah.
But they put out a bunch of software and stuff to do genomics. And so the Solera assembler is like one of the original assemblers for long reads and stuff like that. It's the program that, it all stemmed from the program that he used to assemble the human genome and his team used to assemble the human genome. It's updated to account for the error profile of these long individual sequence trees.
Yeah.
It does, it gives good assembly. It's a good assembly.
Yeah.
If anybody wants to use it. Yeah, I think.
Yeah.
Thank you, Bridie. Welcome.